This is a production of Cornell University.
Thank you very much, Ian. After all that administration, I'm pleased to tell you I'm doing very little now, and I'm doing a lot more science, and it's a lot of fun. And one of the things that I've always found fun is my studies of isoprene. And so I'd like to cover a lot of things, so most of them won't be in great depth, but I hope it'll tell an interesting story. I wanted to mention or talk about global warming, and I'll show some of the requisite slides just so we're all on the same page about CO2 and temperature, and then talk about how these affect photosynthesis, just as a background on which we'll layer isoprene. And I'll talk about isoprene's role in the atmosphere. And then these days we have to call everything omics. So I'll talk about phenomics, genomics, transcriptomics, and phosphoproteomics. But I hope that it will be an interesting story. I wanted to do a little bit of history about global warming. And in particular, savant Arrhenius. But if we look at the history, Fourier, who was a mathematician and was behind the Fourier transform analysis, if you've done any of those, said that the atmosphere contributes to planetary temperature. That if you do calculations, a planet this far from the sun should be so cold as to not support life. And so why does it? Why is it warmer? And Fourier said, well, it's the atmosphere. Eunice Foote observed that different gases had heat trapping effects. Tyndall, that we're going to see later, describes how CO2 is blocking specifically infrared. But Arrhenius calculated warming from a doubling of atmospheric CO2. And this metric stayed with us for a long time. So many high CO2 experiments are doubling CO2. And when I started, the CO2 concentration was 330 ppm. And so doubling it was going to 660 ppm. Now the CO2 concentration is almost 420. So doubling means going to 840. So the experiments are a little different these days.
Well, any questions?
Yeah, sure.
So this Saussure, who is a photosynthesis guy, measured the CO2 concentration in the Alps, but you don't have his name.
No, this is not my history.
Yeah,
I just pulled it out.
History,
It's incomplete. Absolutely.
theory.
Yes, yes. But I wanted to particularly point out Arrhenius. Now, Ångström said, you know, Arrhenius's analysis is not correct because there's too much water in the lower atmosphere, and that would make it blind to CO2. And Arrhenius came back and said, no, the upper atmosphere is dry. CO2 is going to be a problem. So if you have these kinds of problems, these kinds of disagreements, you simply do the experiment. And that's what we've done. The CO2 concentration for here, 800,000 years, between 200 and 300 ppm. And then now we're up at 420 ppm. And indeed, it causes the temperature to go up. These are very clear things that we're living with today. A couple of other things that aren't apparent in these graphs is that warming is greater over land than over oceans. So if you talk about a 1 degree C global increase, you're talking about 2 or 3 degrees C over land areas. So it's really quite a bit more. Another thing is that plants experience the actual temperatures, not the average. And so when you put more energy into the atmosphere, then you get more extreme events. So you have more times when temperatures are extremely high. And you don't capture that if you just simply report averages. And the other thing is then heat waves are more common and intense in a high CO2 world. So I want to talk about how these two things, temperature and CO2, affect photosynthesis. And then, or actually first, I want to show you the things we do to analyze temperature responses. And you can use arbitrary equations, as June and Evans and Farquhar did, to describe, in this case, described electron transport as a function of temperature. And they just used an arbitrary equation. And there's nothing wrong with that, actually. People often use this Q10 analysis. And the nice thing about that is that it's very intuitive. It simply says, if you increase temperature by 10 degrees, what happens to the rate of what you're looking at? Typically in biological systems, it doubles. But it doesn't describe the fall off, the Q10 analysis. If you want to be a little more formal, we use the Arrhenius equation. So this is the same Arrhenius that talked about doubling CO2. The rate is equal to some pre-exponential term times the exponent of this EA is energy of activation divided by gas constant and temperature. And what people do with that a lot is to plot, make an Arrhenius plot. You take the natural log of both sides of this equation, and you get very nice straight lines, and you can calculate then this energy of activation, and you can compare processes. Okay, does this have a higher or lower energy of activation? But a more complete equation is the Eyring-Johnson equation. And the nice thing about that is that it does account for the decline at high temperature. It introduces more variables, the delta HA, it's enthalpic energy and energy of activation, and introduces delta S. So those are the ways, and we'll see examples of those throughout the talk. So when we look at photosynthesis, it responds to light, and then there's a saturation point where it doesn't respond quite as much. Photosynthesis response to temperature is more modest than most people think, and I'll show you some of the reasons for that. And then the response to CO2, we know that it responds strongly to CO2, and then it slows down. So the response to CO2 is made up of three components. One is just the properties of Rubisco. So you assume that RUBP, the substrate, is made at faster rate than Rubisco can use it. So you've got RUBP saturated Rubisco, and that would give you a CO2 response that looks like that. Can predict very well down here, but it doesn't predict well up here. You can assume that electron transport limits photosynthesis. You overpredict here, but you catch these points real nicely, but then you overpredict here. And this is a third thing we call carbon output. What happens as you fix CO2, you've got to somehow deal with the sugar that you've made.
So
yes?
we cannot see your pointer on the screen.
You can't see my pointer. Thank you for telling me. Let's see if I.
Oh, yeah.
Okay. The arrow was there. Thanks. Okay, so now, why is it that photosynthesis is less sensitive to temperature than you would expect? Rubisco is an enzyme. So many enzymes have a Q10 of about 2, energy of activation about 60,000 joules per mole. But what happens is if you assume the maximum rate of Rubisco, it has this very strong temperature dependence, just as you would expect. But Rubisco is operating at a concentration of substrate, CO2, that's about the Km for CO2. And then the velocity of the reaction is going to be proportional to the Vmax and the Km divided by Km. And both of them have a strong energy of activation. And so when you take the ratio, it changes this behavior to this behavior here, the middle line. So it makes it much less sensitive to temperature. You may know this process of photorespiration where oxygen is fixed instead of carbon dioxide. And that increases with temperature faster than does carbon fixation. And so that changes that relationship a little bit more. So Rubisco is less temperature sensitive than you normally would think in the plant. People think that electron transport is going to be independent of temperature. And the actual process of absorbing light energy is independent of temperature. But when you look at electron transport, you've got to deal with all of the components of electron transport, things moving through membranes and that kind of thing. And it ends up with looking somewhat like that. And then when we look at carbon output, how fast you take carbon out of the Kelvin-Benson cycle, you get a stronger temperature response, but then a decline up here. And so we use that Eyring-Johnson equation for that. So with that background, I want to talk about plant resilience to global change, and specifically the case of isoprene. It surprises many people, it surprised me, when I learned that oak trees give off more hydrocarbons than pine trees. If you go into pine forest, you're well aware of the monoterpenes being given off by pine trees. You go into an oak forest and you're not aware of the fact that there's even more hydrocarbon, and that hydrocarbon is isoprene. We're relatively insensitive to isoprene. If you're smelling isoprene, you're getting a toxic dose. So do something about that. And the amount that's made by plants, not all plants make it, but some do. And the amount that's made is just incredible. So if we look at the total amount of hydrocarbon, including methane in this case, going into the atmosphere, isoprene is nearly half of that total. The monoterpenes that you can smell and have these pleasant smells generally make up a big component. The things we worry about, the anthropogenic, these are unburned gasoline and things like that, is only this much. So it's surprising that hydrocarbon emissions into the atmosphere are primarily biogenic, primarily natural processes. And they can become a problem. Now, recognizing this, it was a presidential candidate back in 1980. So for people of a certain age, we all remember this. Ronald Reagan said 80% of our air pollution comes from hydrocarbons released by vegetation. So let's not go overboard in setting and enforcing tough emission standards from man-made sources. And when you follow that, you get this kind of thing. And I will say, I don't think it's all the trees in Los Angeles that makes Los Angeles look that way. So what is the issue? Well, when we look at ozone, so what's happening here is you've got ozone and you've got hydrocarbons and you're making particles, and they're scattering light, and they're scattering red light because they're big particles. And so when we look at ozone, we define this NOx, you might hear of NOx, NOx, as the sum of NO and NO2. Okay. So N2 and O2 is actually, would not exist. If you look, if there was somebody way out in space, looked at the Earth and saw evidence of O2 and N2 existing together, they would say there's something wrong on that planet because that's not in equilibrium. Now, fortunately, there's a large energy of activation, and so you don't get that much NOx, but you do get some NOx from lightning, and you get some NOx from cars, a lot from cars, in fact. But the total amount of ozone that you can make is equal to the total amount of NOx that you have if you don't have any hydrocarbons. Okay. These two reactions happen. So at night, all of the NOx is as NO2. During the day, sunlight causes the NO2 to break apart. You have atomic oxygen connects to molecular oxygen to make ozone. Okay. Now, here's where it gets interesting. If you have isoprene or any other hydrocarbon, they will interact, it will interact with hydroxyl radicals, OH, not OH minus, just OH. And skipping a bunch of steps, that leads to NO2 being made. So you're taking some of this NO and you're converting it back to NO2, and you're leaving some of the hydrocarbon behind. So this allows you to make more ozone. And that hydrocarbon that's left behind can recycle, and you can make a lot of ozone from one isoprene molecule. Now, if you do these reactions and the atmosphere is pretty clean, you actually clean the atmosphere with isoprene. If you get a certain amount of NOx present and you add isoprene, then you get more ozone. So the isoprene will contribute to ozone pollution. So the solution is to not put NOx in the air. This was recognized some time ago. Rai Rasmussen advised the EPA that you should not put all your eggs in one basket. You should not try to suppress hydrocarbon. We tried to suppress hydrocarbon emissions from cars, and it didn't work to lower ozone. And the reason is that cars are only a small part of the hydrocarbon emission. Rai was a student of Fritz Wendt, and so many people will remember Fritz Wendt for his work with auxin. But he, later in his life, he was just fascinated with the hydrocarbons coming from, especially leaves on the forest floor. And he published about the Blue Mountains. And it is the case that isoprene contributes to aerosol formation. And you can do this, monoterpenes are even better, so I've got a demonstration with monoterpenes. You fill this container here with ozone, and then you take a lemon peel and you just squeeze it on top, and you can watch the individual droplets go down, because as the hydrocarbon evaporates from the droplet, it forms particles just that fast. And this is something that Tyndall, that we talked about before, had talked about. So hydrocarbons in ozone will make aerosols, and these can be a problem, especially the small aerosols that are made this way, because they can get right through your lung tissue into your bloodstream. So maybe we should just cut down all the trees. I don't think so. But plants aren't the only organisms that make isoprene. Almost everything does.
I was told that I emit isoprene. We are going to test to see if that's true and how much. It looks like approximately 40 parts per billion.
So everybody that I've ever tested has isoprene in their breath. And for many people, it's the most abundant hydrocarbon in your breath. Second is acetone. And we know where acetone comes from, the ketone bodies, but we don't have any clue yet where the isoprene in our breath is coming from. So the next thing I want to talk about is the genomics. If we get the gene sequences for a bunch of isoprene synthases, we can start to figure out the evolutionary history. And we have isoprene synthases here right next, the true isoprene synthases, next to these that at this time we were calling predicted isoprene synthases. I don't say it on here. These are mostly rosids. These are astrids. And the spices clearly went crazy when it came to monoterpene synthases, and that's why we think of them as spices. But they're right next to acyclic monoterpene synthases. And what we discovered over time is that there were a few amino acids. If you change a few amino acids, you convert one particular kind of monoterpene synthase. And this is an osamine synthase. So this is osamine. And you see it's similar to isoprene, it's just a little bit longer. And all of the osamine synthases have an alanine at a particular location, whereas all the isoprene synthases have a phenylalanine at that same location. So that the pocket is smaller in the isoprene synthases, and it will only fit the DMADP. What I don't understand is why DMADP doesn't come in here and why all osamine synthases are not isoprene synthases, but it seems to be the case. But what it means is that it's very easy for isoprene emission to evolve. And so we know that it's evolved independently many, many times now, quite a few times just in the angiosperms, but we find it in lots of different species, and we have no idea what an isoprene synthase in, say, mosses or ferns looks like. We know that the mosses and the ferns don't have any genes that are remotely like the gene for isoprene synthase. So next year, we're going to do a big push with the water fern called Azolla. It's a pretty big isoprene emitter, and it has been fully sequenced. So we're going to go in and see if we can find that gene. All right, so now if we look at phenomics, so this is what we had developed for photosynthesis, and let's look at what happens with isoprene. Isoprene is light dependent. You turn off the light and isoprene emission stops. It will continue to increase with the light even after photosynthesis is fully saturated, and we don't know why that is yet. If we look at temperature, isoprene emission is much more sensitive to temperature. Q10 sometimes of 5, which is just much greater than any other process that I know. And a surprising thing, and I warn you now, I will not be able to answer why, is that isoprene emission is suppressed by high CO2. So I will, over several slides, talk about this difference of increasing with temperature, but decreasing with CO2, which is going to win out. What will happen in the future? Will there be more isoprene or less? So this is the data about the light response of isoprene emission. Francesco Loretto was with me for 5 years and is doing great work in Italy now, collected this data. This was pretty extreme, but many, many people have found this, that isoprene emission continues to increase with light after photosynthesis is no longer sensitive. Isoprene emission requires a lot of ATP and requires a lot of NADPH, so this kind of makes sense. When we look at temperature effects, this red line is the temperature of the chamber. So that's how fast we could change the temperature of the chamber, or of the, actually of the leaf itself. And this is the rate of change of isoprene emission. And we see that there were two components to this. So if you do a log plot, where you have this break, you have two different components. And we didn't really understand it at the time. And one of the ways we addressed that is to look at post-illumination isoprene emission. So if you turn the light off, isoprene emission goes down to 0. Then it comes up a little bit and goes back down to 0. We call this a little burst or a little burp. And we have to look at how fast isoprene can clear the chamber, and that's this dashed line. What it allows us to do is to get an estimate of the amount of immediate precursor to isoprene and the amount of a precursor that's further up. And we've done the GC mass spec or the LCA mass spec to confirm that the integral of this, the area of this, is directly proportional to the amount of this intermediate. And the integral of this is directly proportional to the amount of these two compounds. And DMADP is the compound that is the immediate precursor to isoprene. Now, I want to show you temperature responses. So as you increase temperature in this range up to 35, it doesn't matter how fast you go. So if you go really fast, the black dots, then you get this curve. If you go, so this is spending 5 minutes at each temperature. This is spending 10 minutes at each temperature, and this is spending 40 minutes at each temperature. So we wanted to know why that is. You can imagine that if you just do a jump from 30 to 40, so we jump from here, 30 to 40, that over time, there's an increase and then a decrease. So that's what we find here, increase and then decrease. And so we looked at the metabolites here, here, and here. And what we find is that when you first increase to 40 degrees, there is a big increase in metabolites. So metabolism goes faster when you go to high temperature, providing more of the substrate. And then that substrate goes away after an hour at high temperature. If we look at the total amount of MEP, so this is those two different areas, we integrate those two different areas and add them together, there's a nice big increase in the total amount of metabolites in the MEP pathway, and then it goes down, declines. If we look at the rate constant for the enzyme, as you go to, as you increase the temperature, the rate constant goes up, as we normally expect for enzymes. And the rate constant stays the same at 40 degrees. And so the reason for this burst is an initial flood of the pathway with metabolites, followed by something that restricts the metabolites. And we don't know exactly what that is. So this is looking at isoprene emission as a function of temperature. If we look at photosynthesis, it declines quite a bit at these temperatures, so that the carbon cost, the amount of carbon going into isoprene at 25 degrees is maybe only 1%. But by the time we get to 45 degrees, we're looking at 20% of the carbon coming in is coming straight back out as isoprene. And that's a function of two things. One is increasing metabolites and increasing activation of isoprene synthase. And if we do an Arrhenius plot, we get a nice straight line telling us that everything is working just fine with the enzyme. Now, those are all instantaneous temperature effects. We also looked at long-term temperature effects. And with Poplar, it's a sometimes when you grow it, it will continuously flush, continuously make new leaves. And so we have, if we look at this developmental scale, we have, we can look at one point and look at the age, different aged leaves. And what we see is that when we study photosynthesis, as the leaves develop, photosynthesis comes up, and it's pretty much the same whether you're developing at 20 degrees or 30 degrees. Later on, 30 degrees has a slightly higher photosynthetic rate. But for isoprene, even after the plants are relatively competent for photosynthesis, the isoprene comes on much slower. At 30 degrees, not that much slower, but at 20 degrees, quite a bit slower. So if you grow plants at different temperatures, plants grown at low temperature have a lower rate, a lower capacity for photosynthesis. So we wanted to study that, going back and forth. And Amy Weiberly did this experiment, where she had plants that had developed at 20 degrees C, measured isoprene. She had plants that developed at 30 degrees C and measured isoprene, always measuring at 30 degrees C. And then she switched from the low temperature to the high temperature, and isoprene emission went up. Okay, that makes sense. She switched from the high temperature to the low temperature, and isoprene emission went up. And in fact, the pattern that we saw was that whenever you make a change, there's an initial increase in isoprene emission that then goes away. In the long term, if you grow plants at higher temperature, they have a higher capacity for isoprene emission. There's not a lot happening with photosynthesis other than initially at low temperature, it was low. But it's relatively insensitive to growth temperature, photosynthesis is, whereas isoprene is very, very sensitive. All right, the next thing I want to talk about is the CO2 response. So here's the CO2 response of photosynthesis. Nice pretty curves. This is photosynthetic electron transport. We can get at that with chlorophyll fluorescence analysis. And then when we look at isoprene emission, it doesn't follow any one of those. We have not been able to correlate isoprene emission to any parameter of photosynthesis. We've looked at a whole bunch of different parameters, and we cannot correlate it with anything. If we look at it at different temperatures, here we see again this incredibly strong temperature response of isoprene emission capacity. This is so at low CO2, we go from 10 to almost 40. So Q10 close to 4. And photosynthesis at these 3 different temperatures, pretty much all the same. Electron transport, pretty much all the same. So none of these things in these 2 help us explain this. So we had this question, in the future, what's going to happen? Will high CO2 suppress isoprene emission, or will the high temperature cause it to be stimulated? And we had an undergraduate student come for the summer, and she tested in 4 different conditions. We have tobacco plants that were engineered by Claudia Vickers to make isoprene. So we have plus and minus isoprene there. We have Phragmites, we have sycamore, and we had poplar. And in the tobacco, there wasn't a significant difference. We measured at 400 ppm at 28 degrees and 800 ppm at 33 degrees because as far as when we looked at the modeling of that, it said that by the time you get a doubling to 800, that there would be a 5 degrees C increase in the temperature that these plants would experience. But for the Phragmites, the sycamore, and the poplar, they all have a higher emission in the future atmosphere. So the temperature response is greater than the CO2 response. So we're likely to get that. Knowing that there's that much isoprene being produced, we ask the question, what does it do for the plant? Why do some plants make isoprene? And so this was one of the early experiments. We gave the plant 3 heat spikes going to 42 degrees Celsius for 2 minutes. This is an oak leaf. The plus means that the oak leaf is still making isoprene. The minus means it's not making isoprene because of an inhibitor, phosmidomycin. And after these 3 heat spikes, we've lost 1/3 of the capacity for making isoprene. But in the absence of isoprene, we've lost 2/3. So it allows the plant to tolerate these heat spikes much better if it's making isoprene. Of course, we want to do it with Arabidopsis. If you give Arabidopsis 20 ppm isoprene, it survives better. Not great, but it survives better 24 hours at 40 degrees C. And some of my colleagues around the world have found the same things, that it protects against this kind of really significant heat stress. We're also looking at protection against ozone. And so Bianca Cerda is giving an ozone treatment to a section of a tobacco leaf and then exposing it to ozone, giving isoprene treatment, then exposing it to ozone. Or she doesn't give it the isoprene pre-treatment, just expose it to ozone. And you can see as the days go on, there's complete destruction of the leaves by this ozone treatment and almost complete protection if they're first exposed to ozone. And this is happening even though there's not isoprene present when there is ozone being given. So you give a pre-treatment with isoprene, and that changes the plant somehow so that it can then tolerate the ozone. We did another experiment, Chris Harvey did this, where he exposed the plant to methylvinyl ketones. So isoprene, the first reaction that breaks down isoprene in most cases, in many cases, is splitting it into methylvinyl ketone and methacrylate. And methylvinyl ketone is what's called a Michael's addition chemical, and it's reactive, causes oxidative stress. And if we take our control Arabidopsis and give it isoprene, we don't change the amount of hydrogen peroxide in the leaf. If we give methylvinyl ketone, we do change. We get hydrogen peroxide buildup. So there's Ross stress. If we give methylvinyl ketone and isoprene, we get no change from the background level. So that isoprene prevents the reaction causing excessive hydrogen peroxide, but it doesn't quench hydrogen peroxide itself. So it attacks the synthesis of hydrogen peroxide. That's what we believe. For a long time, I had pushed the idea that this was related to isoprene dissolving into membranes. And Chris, as a graduate student, dared to contradict me. And he showed that if he takes nitrogen, humidifies it, adds isoprene, and you can't bubble through a solution of vesicles, you get just bubbles. But you let it equilibrate here a lot, and then you let it equilibrate with just the buffer solution. And then you take samples from here and you strip the isoprene out. And what he found is that when there was either thylakoid vesicles or just a model membrane lipid, there was more isoprene. And so this is the isoprene that was in the membranes. So then we can take that amount of isoprene in the membranes and compare it to how much membrane there is. So for every 1 million membrane molecules, there was only about 64 isoprene molecules. So it really is not isoprene dissolved into membranes. All right. So we did some RNA-seq, and we found just an incredible variety of genes that were expressed. We've tried to organize these. One of the clear signals was JA synthesis signaling. So the lipoxygenases and many other things that are involved in JA synthesis. And phenylpropanoid synthesis was affected a great deal. Other things, there was some cytokinin, not a great deal. If we organize it a little differently, again, a small amount of cytokinin signal, strong jasmonic acid signaling. Funny with ABA, there's some signaling increasing and some decreasing. And ABA effects are known to be that way, that there are some ABA effects more connected with drought and other ABA effects connected with other things. The salicylic acid was suppressed, and there's a very common balance between jasmonic acid and salicylic acid. When one is stimulated, the other is inhibited, and vice versa. So Sarathi put these all together. These dashed lines are all effects that we saw as a result of feeding isoprene, and these are all transcription factors. And so they start a number of processes that change growth, and we think change the ability of the plant to tolerate the various stresses that we would throw at it. We did phosphoproteomics, and some of those were surprising. For example, chloroplast movement. Well, I wanted to, yeah. Okay. So CHUP1 and PHOT2, if you know chloroplast movement, CHUP1 connects the cytoskeleton to the chloroplast, and PHOT2 is a blue light sensor. And so there are changes. This is 1 hour of isoprene treatment, followed by 5 hours, followed by extraction and doing the phosphoproteome. But there were other ones that were quite surprising to me. A number of sugar metabolism proteins were changed in phosphorylation status. RNA binding and processing. The PAB4, I think, is involved in these stress granules inside the nucleus. And so we don't know what these things all mean. If you know your membrane protein trafficking genes, some of those were phosphorylated receptors, and then signal transduction pathways like kinase 7 is a very common signal transduction pathway. A lot of it is involved with, connected to this enzyme, which has both a kinase and phosphatase domain. It's a PP2C phosphatase and a kinase, both together. So just more transcription factors. I think in the interest of time, I will just say that we've been trying to put these all together in different pathways. And some of it's making a lot of sense, but it says that isoprene is affecting gene expression and also phosphorylation status post-translational modifications. Just one other observation. We don't know why, but we have Arabidopsis strains that make isoprene and those that don't. And here are seeds of Arabidopsis strains that make isoprene, and they grow longer roots than the controls. But if you grow controls and isoprene emitting lines together, you get the same root lengths regardless. So this is a kind of isoprene signaling from the ones that are making isoprene to the ones that aren't. So if you grow them in separate Petri plates, the isoprene emitting plants have longer roots. Yeah. If you grow them together in one Petri dish, they all have the same length of roots. So that's the story. I wanted to tell you about the role in the atmosphere, the phenomics, genomics, metabolomics. But the role in plants then we think is to do with abiotic stress, especially temperature and ozone. And I think isoprene doesn't quench ROS. A lot of people think that, but I don't think it quenches it, but it prevents it from being made and prevents ozone damage in that way. There's a story about biotic stress, but I'll leave that for another day. So I want to thank you for listening, and here are the people that are working on that now. And thank especially NSF, who's funded this off and on for many years. Thank you.
Thank you so much, Tom. Very informative talk. I learned a lot. Questions? Class?
Yeah, so isoprenes that the plants emit, is it coming from the plastid or from the downstream, the MET pathway and not the MVA pathway?
It's definitely MET pathway.
Why is that? Does it hinge on a specific gene or one gene, or?
There's just one gene. All organisms have DMADP, dimethyl allyl diphosphate. And dimethyl allyl diphosphate will spontaneously break down to isoprene. It's actually a bit of a problem, but nowhere near the rates as in plants. And it's these isoprene synthases. And so we've cloned them a lot and measure them in the test tube. And then the other thing is we feed C13O2, and we see that it labels up really, really fast. Isoprene labels up very fast. And if we inhibit the MEP pathway with fosminomycin, we knock out isoprene emission. So it's definitely in the plastid.
Any other questions? Yes.
And is the action spectrum of isoprene emission the same as the action spectrum of photosynthesis?
Yeah, we haven't done real detailed analysis, but I think so. But are you thinking maybe a disequilibrium between PS1 and PS2 or something?
No, I'm not thinking in detail. I'm just,
Oh, okay.
I loved your seminar and I'm just
Okay.
thinking.
Yeah.
I have another question. I know you talked about the ozone aspect of isoprene, but do you know the scattering spectrum of isoprene?
No, we don't know this. Don't know the spectrum, but we assume that it's just Raleigh scattering, which is just wavelength dependent. The shorter wavelengths are scattered more than longer wavelengths, and that's why
With no peaks?
no peaks as far as I know. But you might know. Does Raleigh scattering have a?
Well, Rayleigh doesn't, but
Yeah.
does isoprene.
Yeah, no,
Like CO2 has.
yeah. Isoprene doesn't absorb in the visible. It does absorb in the UV, but not in the visible. So.
And one more question. Are dolly calls used for sugar transport across membranes in the plastic?
I don't know. I mean, I only know of dolichols in human medicine. I presume that, but I would presume that they're more important in the ER. But we don't know that the effects of isoprene are in the plastic. We know the synthesis is in the plastic, but the effects could well be in the ER. But the dolichols are made from the methylonic acid pathway. So I think I'm pretty sure they are.
Any other questions? Yes.
That was an awesome talk. I learned so much about isoprene. Thank you so much. So my first question is related to the root growth pattern you saw at the very end there. I was kind of curious about whether heard of maybe does the isoprene, the lack of isoprene production, I guess here, would it possibly affect the exudation patterns in the roots there and maybe have implications for soil carbon? Do you know anything about that?
I hadn't thought about that, but it could well, yeah.
Okay.
Yeah.
And the other part of my question was, so you showed that with climate change, you're expecting more isoprene production. I was wondering whether you know of any sort of feedback loops with climate change, isoprene production, and maybe microbial degradation of isoprene or usage or anything like that. Is there anything you've heard of or looking into?
No, I'm not looking into it. There is microbial degradation of isoprene, but it's much less than in the air. It's much less. Microbial effects are much less than the destruction by hydroxyl radicals and ozone itself. But in the soil, the things could be quite different. I just recently established a system for growing plants aeroponically. So hydroponics work well, but the plants don't grow quite the same. And if you grow them aeroponically, you just make a mist that the roots are hanging in. They look a lot closer to the way they look in soil. So we're going to start doing fumigations of the root zone separate from the shoots.
Cool. Awesome. Thank you.
Yeah.
Thank you. Any other questions in the room? Okay, let's take questions from Zoom. There were like more than 70 folks joining on Zoom.
Wow.
So Thomas, thank you for your patience for keeping your hand up. Thomas, go ahead.
Oh.
So hi, this is Thomas Bjorkman. So thanks for coming to Cornell. Good to see the update on this story. So I was wondering, there's not intense work, but there's interest in breeding for increased climate resilience in crop plants, ornamental landscape plants, and possibly even forestry plants for the various manifestations of high temperature injury. And so I wonder whether you see opportunity for using isoprene or breeding for isoprene as a protection for those high temperature events.
Yeah, we argue that both ways. One is we should have plants make isoprene. It would be very easy to make soybeans make isoprene. The precursor to soybeans, glycine soja, makes isoprene and has proper isoprene synthesis. The isoprene synthesis in glycine max seem to be non-functional. So we could make soybeans make isoprene. Now, are we improving things or making things worse? So they're more tolerant of ozone, but they're making isoprene, which contributes to ozone. So we argue back and forth. I don't know what the best thing is, but I think that for me, this is one of the reasons I like to do this research, because as we understand more and more of this, we might be able to answer your question exactly, that is this a trait we should put into crops, or is this a trait we should take out of the plants that we're using?
Yeah, thanks.
Thank you. Any other questions from Zoom audience? Okay. Actually, I have a question related to what Thomas asked. Say, you talk about climate change, that's just the main pattern, but of course more heat waves. So I would assume there would be isoprene emission volcano during those heat waves, and that would probably be much more than what the main warming would induce, right?
That's exactly right. Yes. So if you have a hot spell of 4 or 5 days, each day the capacity for making isoprene would be going up because it's growing in a warmer temperature, and also each day the temperature is warmer, and so they're emitting more of their capacity, a greater. And so I think this is why these heat waves become such a problem for atmospheric, for air pollution, because you're putting in so much hydrocarbon from the isoprene.
Yeah, okay. Yeah, I have another naive question of how, because NPQ, non-photochemical quenching, is another plant protective mechanisms. So are they correlated with each other, or?
Not at all.
Okay.
We tested NPQ, we tested electrochromic shift, we tested photosystem 1, we tested nothing explained it. For a long time, I was sure it was this carbon export process, TPU, and we decidedly proved that was not the case. 10 years of hypotheses down the drain. So.
Okay. Yeah. Thank you very much. Yeah. Okay. So let's thank Tom again for his excellent talk.
This has been a production of Cornell University, on the web at cornell.edu.